Welcome back to channel. Today topic is Centrosome and Centrioles. Nucleus. First topic we entree into Centrosome and Centrioles. Centrosome is an organelle usually containing two cylindrical structures called centrioles. They are surrounded by amorphous pericentriolar materials. Both the centrioles and a centrosome lie perpendicular to each other, in which each has an organization like the cartwheel. They are plasma membrane peripheral microtubules interdoublet bridge. Central radial microtubule spoke central sheath. Cilia/flagella showing different parts. A. Electron micrograph diagrammatic representation of internal structure. Made up of 9 evenly spaced peripheral fibrils of tubulin protein. Each of the peripheral fibril is a triplet. The adjacent triplets are also linked. The central part of the proximal region of the centriole is also proteinaceous and called the hub, which is connected with tubules of the peripheral triplets by radial spokes made of protein. The centrioles form the basal body of cilia or flagella, and spindle fibers that give rise to spindle apparatus during cell division in animal cells. Next topic is nucleus. Nucleus as a cell organelle was first described by Robert Brown as early as 1831. Later the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming. The interphase nucleus of a cell when it is not dividing, has highly extended and elaborate nucleoprotein fibers called chromatin, nuclear matrix, and one or more spherical bodies called nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 50 nm, called the paranuclear space, forms a barrier between the materials present inside the nucleus and that of the cytoplasm. The outer membrane usually remains continuous with the endoplasmic reticulum and also bears ribosomes on it. At a number of places the nuclear envelope is interrupted by minute pores, which are formed by the fusion of its two membranes. These nuclear pores are the passages through which movement of RNA and protein molecules takes place in both directions between the nucleus and the cytoplasm. There is only one nucleus per cell. Variations in the number of nuclei are also frequently observed. Can you recollect names of organisms that have more than one nucleus per cell? Some mature cells even lack nucleus, e.g., erythrocytes of many mammals and sieve tube cells of vascular plants. Would you consider these cells as living? The nuclear matrix or the nucleoplasm contains nucleolus and chromatin. The nucleoli are spherical structures present in the nucleoplasm. The content of nucleolus is continuous with the rest of the nucleoplasm as it is not a membrane-bound structure. It is a site for active ribosomal RNA synthesis. Larger and more numerous nucleoli are present in cells actively carrying out protein synthesis. You may recall that the interphase nucleus has a loose and indistinct network of nucleoprotein fibers called chromatin. But during different stages of cell division, cells show structured chromosomes in place of the nucleus. Chromatin contains DNA and some basic proteins called histones, some non-histone proteins and also RNA. A single human cell has approximately 2-meter-long thread of DNA distributed among its 23 pairs, chromosomes. You will study the details of DNA packaging in the form of a chromosome in class 12. Every chromosome, visible only in dividing cells, essentially has a primary constriction or the centromere, on the sides of which disc-shaped structures called kinetochores are present. Centromere holds 2 chromatids of a chromosome. Based on the position of the centromere, the chromosomes can be classified into: the metacentric chromosome has middle centromere forming 2 equal arms of the chromosome. The submetacentric chromosome has centromere slightly away from the middle of the chromosome, resulting into 1 shorter arm and 1 longer arm. Acrocentric chromosome, the centromere is situated close to its end, forming 1 extremely short and 1 very long arm, whereas the telocentric chromosome has a terminal centromere. Sometimes a few chromosomes have non-staining secondary constrictions at a constant location. This gives the appearance of a small fragment called the satellite. Microbodies. Many membrane-bound minute vesicles called microbodies that contain various enzymes, are present in both plant and animal cells.